Hello everyone, welcome to today's live broadcast, Humanized NSG Mice for Evaluation of Immuno Oncology Therapeutics. I'm Judy O'Rourke of LabRoots and I'll be your moderator for today's event. Today's webcast is presented by labroots.com, the leading social media site for science professionals, and sponsored by the Jackson Laboratory. The Jackson Laboratory is a leading provider of cancer mouse models and in vivo oncology services, and it is a National Cancer Institute designated cancer center. Drawing on decades of mouse-based oncology research experience, the Jackson Laboratory provides capabilities around model selection and husbandry and customizable oncology studies to help clients and researchers evaluate novel anti-cancer therapies. The Jackson Laboratory's diverse oncology study protocols range from traditional xenograft services to patient-derived xenograft cancer models and advanced humanized mouse models for immuno-oncology drug development. They have the flexibility to customize studies and import client-provided mice and cancer cell lines. For more information, please visit www.jax.org. Before we start, there are a few instructions. We want to hear from you during this interactive presentation, so please ask questions or leave us a comment. Answers welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can and we'll follow up if we don't have time today. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button at the top right or use the Q&A button. We'll make sure we try to resolve any issues. Now let's get right to today's presenter. We are proud to welcome Brian Soper, PhD, Technical Information Scientist at the Jackson Laboratory. Dr. Soper has worked at the Jackson Laboratory for 20 years. He conducted research on treatment strategies for a mouse model of human enzyme deficiency. Approaches included enzyme replacement, gene therapy, and neonatal allogeneic bone marrow transplantation. As a technical information services scientist, his area of expertise is in modeling and complications associated with human type 1 and type 2 diabetes and research with immune deficient mice. I will now turn it over to Dr. Soper.
Okay, wonderful. Thank you. Thank you for everyone for joining us. I'm going to go over a few different things. Let's begin with a presentation outline. We're going to begin with an overview of the immunodeficiency of the NOD.Cg gamma mouse so you can understand in detail why this is such a more important animal and why it makes itself amenable to the great research that we're going to be providing you information on today. Then we'll talk about the humanization of that animal, and this is in the context of transplanting human hematopoietic compartment into the mice. We'll talk a little bit about the human immune function and what's known, and then we'll talk about the application of patient-derived xenografts in the NSG, and then we're going to combine the humanization with the patient-derived xenograft platform and talk about immuno-oncology in the humanized mice. So let's begin with an overview of the immunodeficiency of the NSG mice. So what's very important about this model is the genetic background. The genetic background contributes a great deal to this model. It's called NOD, which stands for non-obese diabetic. This genetic background is actually a mouse model in and of itself without the mutations that I'll explain in just a moment. It's a mouse model of autoimmune type 1 diabetes. However, if you put these other alleles that we're going to talk about onto it, it becomes immunodeficient and no longer develops the autoimmune dysfunctions. So it has an absence of hemolytic complement, reduced dendritic and macrophage cell function. It doesn't mean that it's devoid of these cells. The cells are there, but if you compare their function to a BLAC6 or a BELC, for example, they just don't have the same level of response. Now, another allele that the genetic background contributes that's very important is an allele called SERP alpha. SIRP alpha will engage CD47 on the donor cells and tissues that are being transplanted into the mouse. Now, if you were a BLAC6 or a BALB/C, those SIRP alpha alleles would cause the macrophages in those animals to be activated and phagocytose those human cells, preventing good engraftment. However, in the NOD mouse, the SIRP alpha allele does not engage them in a way that activates the macrophages, so this acts as a don't eat me signal. So it makes these mice more permissive for the receiving of these xenogenetic cells and transplants. Then, of course, there's the SCID mutation that has been bred onto this genetic background. SCID stands for severe combined immunodeficiency and prevents the development of mature T cells and B cells. This is a DNA repair site of VDJ rearrangement. VDJ rearrangement or recombination is necessary for the functional maturation of T cell and B cell receptors. So if you knock out that rejoining aspect of that VDJ rearrangement, those cells cannot become functionally mature. The next mutation that was bred onto it was the IL-2 receptor gamma chain mutant. This is a complete null of the common gamma chain. The common gamma chain is part of multiple cytokine receptors. These include interleukin 2, 4, 7, 9, 15, and 21. And many of these receptors are very important for blocking a natural killer cell activity. You may be familiar with a regular NOD/SCID mouse without the IL-2 gamma chain mutation. Those mice still have natural killer cells and serve as a very poor recipient for hematopoietic engraftment. Now, because the SCID mutation is on the DNA repair side of VDJ rearrangement, if you're putting tumors into the flank of these mice and testing chemotherapeutic agents, the host animal itself will be sensitive to those genotoxic drugs, so keep that in mind. So if we go back and we look at the different arms of immunity just to understand all the aspects of immunoincompetence in this mouse, we have the two arms. We have the innate arm and we have the adaptive arm. And if you look at the NOD/SCID gamma mouse, if we look at all the areas that are lightened in this model, we can see that there's multiple immunodeficiencies in these animals. The complement system, macrophages, natural killer cells on the innate arm, and on the adaptive side, we have the B cells, the T cells, inability to make antibodies, reduced dendritic cell function, and then of course, because of the IL-2 gamma chain, we have cytokines that are dysfunctional in terms of the cytokine receptors, which are the crosstalk between the innate and the adaptive arms of immunity. So let's go through now, moving on, now we understand the host mouse. Let's talk about the humanization of the NSG animals itself. So if you are familiar with the literature of humanization of the mice through this methodology, you'll see that the original publications by Dr. Schultz and Dale Greiner et al., they used neonatal recipients. The neonatal recipients were injected intracardiacally or intrahepatically. Now, as you can imagine, this makes it a challenge in terms of colony management. You have to do timed pregnancies, producing neonatal mice, injecting those animals within 24 to 48 hours after birth, and then returning those animals to the mother, hoping the mother will accept them. Now, what we've done in our in vivo services group is modified that to a more simple platform where we take 3-week-old animals and simply do an IV tail vein injection so that we get all of the animals that are going to survive and become engrafted with the cells. As you can imagine, we're isolating the most primitive stem cell compartment, the CD34 positive cells. And so all of the mature cells are missing, and so you put in these primitive hematopoietic stem cells and they need to differentiate out into the periphery. So it's going to take 12 to 15 weeks before those cells are detectable in the peripheral blood in those animals. And we can validate that by flow cytometry. Now, just to set you up, for those of you who are not familiar with all the different aspects of hematopoiesis and the different cellular markers, I'm going to show you this diagram very briefly here. So, of course, in the bone marrow contains the developing hematopoietic compartment. Within the marrow space, along the periosteal surface, you can isolate the hematopoietic stem cells. And those are recognized by a cell surface molecule called CD34. Now, within that compartment, there are some cells, like downstream progenitors, that also express CD34. So basically what you're doing is you're enriching for the stem cell compartment if you positively select for those cells. But you'll notice that the other thing that you'll do is you'll also eliminate other cells like these mature myeloid cells and mature lymphoid cell compartments. And that's a good thing, because you don't want functionally mature T cells in there, because those functionally mature T cells can recognize the host as foreign. But what you can also do is use these CD34 markers as a way of validating that you have the development of these downstream lymphoid and myeloid cells generated from the primitive hematopoietic stem cell compartment. And the hallmark for getting the stem cell engrafted is multilineage engraftment. So taking a look at this paper here, one of the earlier papers on the CD34 engraftment in the NSG mice, we're looking at multilineage engraftment. So we're using a series of antibodies to label cells from the peripheral blood, bone marrow, and spleen of these animals, which are conjugated with fluorescent molecules. And we can look in the bone marrow, the spleen, and the peripheral blood. And what we can do is look for a panleukocyte marker called CD45. So we can get total engraftment in these different compartments by looking at human CD45 versus mouse CD45. And then we can ask, within that human CD45 compartment, how many of those cells are T cells, B cells, and myeloid cells. So in these diagrams here, we're looking at the different cell populations, and each one of these dots represents an individual mouse that's been engrafted. So overall, we see high-level engraftment in all three of these hematopoietic compartments, and we have representative cells from both the myeloid and lymphoid compartments, indicating that there's multilineage repopulation in the recipients. Now, the other thing that I'd like to point out very briefly in the next series of slides is that there is slightly differences from one donor population to the next. So it's very important that if you set up a cohort of these animals, that you validate them, and you validate them for total level of engraftment, because there are going to be slight differences. So the next series of slides is looking at the different hematopoietic compartments, and we're going to look at the bone marrow, spleen, and the peripheral blood. And then on the x-axis, what we're going to do is we're going to look at the different populations of cells. And as you would expect, there's going to be different levels of repopulation for different cell types depending on which compartment you're looking at. And of course, in the bone marrow, you're going to see mostly myeloid cells and less lymphoid cells. So if we go to the next slide, we're going to look at the spleen. And as you would expect, you're going to see a lot more lymphoid cell engraftment in this compartment. And then finally, in the peripheral blood is also going to be more of the lymphoid cells, but you're also going to see the myeloid cell compartments represented as well. So let's talk about the immune function in these animals. So again, we're going to break this down into both innate immunity and adaptive immune functions in these recipients. So let's look first at the myeloid cell compartment. If you take the engrafted CD34 mice and you can pull these human cells out of different tissues like the lung and the bone marrow, and if you put them in liquid culture and expose them to fluorescently labeled beads and ask, can those cells phagocytose those beads? You can, particularly if you increase the temperature to 37 degrees Celsius. You can see that these cells will take up these fluorescent beads, whether or not they're derived from the lung or the bone marrow. And then you can run these cells for a flow cytometer. They'll be fluorescing. You can also look at them microscopically, and you could look at those cells close up and see that they phagocytose those beads. Now, the other thing you can do is you can do an in vivo assay, and you can look for the production of pro-inflammatory molecules after injecting the mice with lipopolysaccharides. Lipopolysaccharides are molecules on the cell surface of bacteria that can be isolated, and you can inject those into the bloodstream of the animals. And then you can test for human-specific pro-inflammatory molecules. And as you can see here, there's a number of human-specific pro-inflammatory molecules that are progressively produced over time after you inject those mice. And it's very important to note that these molecules are interacting with toll-like receptors, TLR2 and TLR4, which are found on the surface of these myeloid cells. And certainly these cells are expressing TLR2 and TLR4. And then as a functional assay, you can take these myeloid cells that have been isolated from these mice, put them in liquid culture, expose them to interferon gamma, and you can see these myeloid cells actually phagocytose and kill salmonella bacteria in the liquid culture assay. So that they do have functional myeloid cell compartment. So the next interesting question in a very popular cell population are the natural killer cells. Now, there's a little bit to understand about the natural killer cell compartment within these mice. Yes, those cells can be found there. You can see them at about 1 to 3% in the spleen and about 7% of the lung. But if you'll notice the markers, they're CD3 negative, NKp46 positive, but the majority of those splenic NK cells are going to be CD56 negative. CD56 is a marker for functionally mature natural killer cells. So there is a limitation. These natural killer cells are there, but they may have some limitation in their function, in their functional maturity. Now you can get these cells to become functionally mature by introducing things like IL-15, IL-12, and poly-IC treatment. And what we're showing here on the right is an in vitro assay where target cells mainly like the K562 cells, or CEM cells. These are cells that express markers that are recognized by natural killer cells. And what you could do is put them in liquid culture along with the natural killer cells, and you can introduce other molecules within that liquid culture like poly-IC, interleukin-12, and IL-15. And you can measure the supernatant for things like interferon gamma production or production of a marker called CD107. CD107 is a marker for degranulation, which is an indication that these cells are attacking and killing these cells. And what you'll notice first here is if we look at the red arrows that I'm showing here just to the left of the box, is that with media alone, you can get some indication that the natural killer cells are able to attack the K562 cells and the CEM cells. But the ability of these cells to recognize and kill these cells is enhanced. If you look at the area next to the blue arrows, you'll indicate that this indicates that interleukin 12 and 15 combination, poly-IC, enhances the upregulation of CD56, and you get enhanced killing of those cells. Here's an indication that there's very robust T-cell activity in these mice. So if you isolate the T-cells from the spleen of these engrafted mice, and then isolate clonal populations, take these clonal populations of CD4 and CD8 T-cells and expose them to target cells that are loaded with chromium. So this is a chromium release assay, and the T-cells are mixed with these chromium-loaded target cells at different ratios as shown by the x-axis down here. And so if the T-cell is killing the target cell, you're going to get release of chromium, which is what we call a percent cytotoxicity. Then what you can do is ask questions about the specificity of this killing by mixing in antibodies. Now, CD4 T cells recognize their targets in the context of MHC class II molecules. So if you add an MHC2 molecule, an antibody that blocks MHC2 molecules, you should see a reduction in killing. And indeed, you see a reduction in killing within the CD4 clones as shown here. If you include an anti-CD1, an anti-class 1 monoclonal antibody, you don't have any reduction of killing. And if you have syngeneic targets included in, a syngeneic target, a non-allogeneic target, you shouldn't have any killing, and indeed you do not. And the corollary is true for the CD8 T cell clones. CD8 cytotoxic T cells recognize their targets in the context of MHC class 1 molecules. So you add antibodies that block class 1, you don't see any reduction in killing. You add class 2 molecules, etc. So what you see is specific killing by these different T cell populations. And of course, if you also sensitize the mice with DNBF, which is dinitrophlorobenzene, you can do 2 sensitizations of these mice and then come back in with a challenge after sensitizing the immune system. you can get a pro-inflammatory response in the ear pinna, and this is indicated by a pro-inflammatory response here. And if you add agents like hydrocortisone, then you have a decreased swelling, indicating that you have delayed type hypersensitivity responses, and you can test for immunomodulatories for those. Okay. Now, one thing that I would like to point out, which is a little bit of a challenge with the model, which is an active area of research, is that there are lots of B cells that are being produced in these mice, but their overall B cell responses are somewhat limited. And this is shown here. B cells, when challenged, will initially make an IgM response. They need to form germinal centers where they can interact with follicular dendritic cells and other important molecules for their functional maturation to become plasmacytoid or long-term memory B cells, undergo class switching, and produce a lot of IgM and other Ig molecules like IgE and IgA. These mice, as shown here in the graphic, have very good IgM production, but have very limited IgG production, as shown here in the gray. Okay, so that is one limitation of the model. In other words, you cannot immunize them and make lots of human-specific IgG long-term stable antibodies. Okay. So let's talk a little bit about patient-derived xenograft testing in these animals. And this would be, first, I'll introduce this in the context of non-humanized NSG mice. What I'd like to point out to you is the Jackson Laboratory has created a large database of these patient-derived xenograft models. Essentially what we're doing is we're taking patient-derived tumors that are removed surgically. Within 24 hours, they're placed into the NSG animals. They're placed into the flank of the animal. If they grow, then we harvest those tumors and we collect a bunch of genomics data on them, gene expression profiles, we collect a library of information about the patient history, etc. And what is showing in this graphic here is a distribution of the different models that we have. We have a number of different models, and so what I'm going to show you next is how to take a look at some more information about those. This is a mouse tumor biology database that we have up on the web page. You can get through this. You can get access to this by something called the Mouse Genome Informatics website. And there's a mouse for tumor biology search for mouse tumors. But in the lower corner of that, there's a place where you can click on, as shown by the red box here, that leads you out to the PDX model search, which is the patient-derived tumors. And this is the interface that you're going to see here. You can search this database by tumor ID if you know it, primary tumor type, the diagnosis, gene variants, if you're looking for specific tumors with specific genetic modifications and mutations, etc., you can do that. And also, there's other ways you can search by indication, etc. If you search by bladder cancers, for example, you get a long list of bladder cancer models here. And if you select one specifically based by the tumor model number, that will take you out to an individual page where if we have standard of care treatment, growth characteristics, and other genetic data associated with that tumor model, you can access it here. So one example of is gene expression profiles. We've looked at a number of genes. The genes are compared to one another in terms of generating a Z profile, so you can look at high expressing and low expressing genes. You can also look at other genes in terms of their high expressing genes, down expression, copy number variation, things like that. So there's a wide range of genomics data that you can find with regard to these specific tumors. So what I'm showing here is a little bit of information about maintaining the tumor heterogeneity. Because one of the advantages of the PDX models is that they are heterologous tumors. And we're trying to keep these at very low passage numbers so that they resemble the original patient-derived tumor as much as possible. So if we look in the upper corner here, we have the tumor model number, TM00186, but we also see PT that indicates the patient tumor histology. So this is prior to engraftment into the flank of a mouse. P0 is the histology of the tumor after being passaged once. and then we have the P1 tumor and the P2 tumor. And essentially what this is showing you is that the tumor maintains its overall histological appearance over passage in the tumor. Now, what's interesting is this particular tumor has a specific KRAS mutation, and it's normal for the epidermal growth factor receptor. And this genetic combination would predict that this model has a specific reaction to known standard of care drugs. Specifically, it would be sensitive to cisplatin and resistant to the drug erlotinib. And so if you put this tumor in the flank of the mouse, and you treat these mice with these agents, you will see that in black we see that this is the vehicle control group. Excuse me. This is the vehicle control. This is the erlotinib. This is the tumor volume over time treated with erlotinib, and you see that there's no regression of the tumor. And if we treat those tumors with cisplatin, you see what we call stabilization of disease in this model, which indicates a very robust response to that chemotherapeutic. Now, just to show you one more example of that, this is a particular patient that showed acquired tyrosine kinase inhibitor resistance. Now, what's being shown in this graphic here is that the flat line shown here, this is a time course of tumor development in the patient. Each one of these bumps that you see here is an increase in tumor burden. So this patient had high tumor burden, and they were placed on erlotinib, and the patient responded quite nicely, which showed a regression of tumor volume in the patient. Unfortunately, this patient also became resistant to that standard of care treatment, and tumor burden returned. The patient went to the clinic. Some of that tumor was collected and was passed on to our patient-derived xenograft library, and that's when we received the tumor. The patient went on to receive a dual therapy, and you could see that this patient was treated with this dual therapy and then became responsive, fortunately, to that therapy. So what we did is we took these tumors and placed them into the mice and to see if they would respond identical to the original patient-derived tumor. They were resistant to erlotinib and responded to the dual therapy of afatinib and cetuximab, shown here. So what the take-home message of this is that these tumors very nicely recapitulate the responses to chemotherapeutic agents the same way that they do in the clinic. Okay, so let's move on to our most recent data. And this is using Immuno Oncology in the humanized mice. And this is combining the humanized mice with putting tumors on board to develop a humanized tumor-bearing mice. So you humanize the mice, as we've talked about already, using CD34 hematopoietic stem cells. Then we put a tumor on board, and this is the tumor-bearing humanized animals. Okay. So what was interesting is the original thought was, if we talk to everyone within the immunological community, the original thought was that, well, if these tumors are not matched, and you do have a functional immune system in these mice, the HLA mismatching, the functional immune cells are just going to reject the tumors, and the tumors are going to be wiped out by the immune system. So one of the first questions we wanted to ask was, well, what is the influence of HLA matching and mismatching in these mice? Does it make any difference, and can the tumors grow? The other thing is, if we could get these tumors to grow in these mice, would they respond to chemotherapies like they did previously, or would this change how they functioned? And if we do get them to grow, will they respond to standard of care checkpoint inhibitors? And so I'm going to show you next the data that we have accumulated with regard to all of these questions. Okay, so the first set of data that I'm showing you here is a comparison to non-humanized NSG mice, shown in green, to the humanized mice, shown in the red squares. So it's green circles and red squares. And we're looking at the humanized mice for this non-humanized mice, and we have 3 different patient-derived tumors. We have a breast, a lung, and a soft tissue carcinoma, and we're just simply comparing the growth characteristics of these tumors in these 2 different platforms side by side. And the overall take-home message of this is that these are non-HLA matched between the donor hematopoietic cells and the tumors. We made no attempt at matching them whatsoever. And we have 100% tumor take rate in both of these models, and the growth rates are very, very similar. And so we were very surprised by and very excited by this data. So the next question is, well, what's going on here? Are the immune cells that we've established in those humanized mice entering those tumor sites? So we also harvested those tumors after we did these growth characteristic studies and harvested those hematopoietic cells and stained for those cells by flow cytometry. So one very important question also answered here, and you should be aware of, is that if you take a patient-derived tumor, the original patient-derived tumor can have passenger leukocytes that go along with that tumor. But as you passage those tumors within mice, they will lose those passenger leukocytes completely. So if we look at the green circles shown here, and if we stain these different tumors for CD45 positive cells, we will see that the non-humanized mice, the tumors pulled back out of those non-humanized mice, there are no human-specific CD45 cells in there at all. But if we look at the humanized mice, there are indeed human-derived lymphocytes, etc., within those tissues. And so these T cells are actually going into the tumor sites and are being rendered anergic. Okay, so the next question we wanted to ask was, as I said, do they respond to standard of care treatments? And this is a tumor. This is a excuse me, I forget which kind of tumor this was, that was placed in the animals, and this is a carcinoma, and it was treated with a 5-FU and Avastin, and then asked if we could get a regression of tumor growth. And indeed, we do see a regression of tumor growth, and we got a statistically significant reduction in tumor volume using the 5-FU. So the tumors can respond to standard of care chemotherapeutic agents. Here's yet an additional study, and this is an additional study where we looked at the response of a breast carcinoma. This is a triple-negative breast cancer, TNBC, breast cancer number 1126. And in this particular case, we treated the mice with either cisplatin or an immunomodulatory drug called Keytruda. Keytruda is an antibody that binds PD-1 on T cells. Now, when PD-1 binds PD-L1, its receptor on cancer cells, that is an immunomodulatory pathway which shuts down the activity of those T cells. And this particular antibody called Keytruda, when you flood the system, will bind to PD-1 on the T cells, block that immunomodulatory pathway, and allow those T cells to become active and lead to tumor regression or cytotoxic killing of those cancer T cells. And so what we're showing here is the vehicle-treated control in the black. They treated in the red here, and then down here is the cisplatin-treated animals as well. So we've got standard of care response, and we've also got indication of immunomodulatory drugs showing a blockage of tumor growth in this animal. So we also looked at the tumors within these mice to see, are there actually human-derived T cells entering these tumor sites? And yes, of course, we've got total human CD45 cells as compared, what we've got is the vehicle control group compared to the treated group. In this particular experiment, there was not enough tumor volume left in the cisplatin-treated group to actually look for the T cells within those tumors. So we're looking at total CD45, so total human lymphoid cells within the tumor site itself. And then we looked at, within that CD45 compartment, what kind of T cells and other cells were there. And we found that we had human-derived B cells, CD4 positive T cells, T helper cells, and cytotoxic C cells, all found within the tumor sites of these mice. So what we also wanted to do was take a look at whether we could use combinatorial therapies to get a response in these animals. So in this particular experiment, we're looking at a lung patient-derived tumor, and we're looking at a combination therapy of Keytruda and docetaxel. Now, unfortunately, we didn't really get a combinatorial effect. If you look at this data initially, it looks like the combination therapy, docetaxel plus Keytruda, as shown here by my arrow, it looked like there was a greater prevention of tumor growth. Unfortunately, what you also will notice in the Keytruda alone arm is that there's a lot of standard deviation is indicated by the error bars. And that's because there was one outlier animal in that group. There was one animal that did not respond to Keytruda in that group. There's an N of 10 in all of these study arms. So if you take out that one non-responder, you'll see that the two lines will overlap one another. So what we're doing now is looking at further studies where we're trying to get the minimal effective dose, and so we can look at some kind of a combinatorial dose response. Okay, but if we look in these tumor sites once again and we look for human-derived hematopoietic cells, we can see that in all of these cells, all of these tumor sites, regardless of treatment, whether it's vehicle alone, Keytruda alone, or the combinatorial therapy, we see similar frequencies of human-derived hematopoietic cells entering those tissue sites. Now, if we take a closer look and look at the histopathology of these, and we stain for human CD45 in green, human CD8 T cells in red, and cytokeratin, which is a marker for the cancer cells, human-specific cancer cell tissues within these sites with these lung-derived tumors, we don't see very many CD8 T cells within these cytokeratin-positive regions at all. So we think that the T cells are kind of in the stroma or in the margins of the tumors. But this is the vehicle-treated controlled tumor group. Now, interestingly enough, in the Keytruda-treated tumors, and you look at the red areas, it looks like what's happening to us is that the CD8-positive cytotoxic T cells are moving from the stroma areas or from the periphery of the tumor and can be found directly associated with the cytokeratin-positive regions of the tumors. So the T cell numbers don't appear to be changing, but the location of those T cells in the tumor sites do seem to be changing. And so we're going to be collecting some more data to specifically address actual T cell numbers versus just percentages of human CD45 cells within the tumor sites. So one of the other questions that we've been asked is, well, the myeloid compartment is a very important factor. There's myeloid-derived suppressor cells, there's tumor-derived suppressor cells called TAMs, there's a number of different cell populations. Obviously, the innate immune system has strong interactions with the with the adaptive immune arm, and people are also interested to understand the role that the myeloid cells play in these the tumor microenvironment. Myeloid cells can produce growth factors to stimulate growth of the vascular system, etc. So one of the things we've started looking at is a new platform for humanization called the NSG-SGM3. SGM3 stands for mice that also, which are essentially NSG mice, but they also have a transgene that expresses stem cell factor, granulocyte macrophage colony stimulating factor, and interleukin-3. So they have an IL-3, it's called commonly stimulating factor 2, which is also known as GM-CSF. And then they call it KIT ligand, but KIT ligand is also known as stem cell factor. And these are human-specific cytokines that can enhance the growth and proliferation of the myeloid compartment mast cells and the primitive human hematopoietic stem cell compartment. So what we did first is just compare the engraftment kinetics of these two different platforms side by side. So we have female NSG mice and we have female NSG-SGM3 mice, as we call them. The mice were irradiated to prepare them for the engraftment of these cells. You need to irradiate these mice, which gives a competitive advantage by ablating the remaining mouse cells in the bone marrow compartment, and gives those donor cells a competitive advantage for engrafting and proliferating out into the peripheral lymphoid organs in the mice. We dosed the mice from cells obtained from the exact same donor. All of the mice got about 130,000 cells. And then we collected blood at different time points post-engraftment, 2, 6, 9, 12, 15, 18, etc. weeks after engraftment, and looked for different markers. Okay, so what we're showing here is we're showing that the SGM3 mice are shown in the red squares, as shown here. Oops, excuse me, let me change my marker. Okay, so this upper graph is the SGM3 mice, and we're following total human CD45 engraftment over time. So the x-axis is weeks post-engraftment. And what we're seeing here is that the SGM3 mice have a more robust expansion of the human donor-derived CD45 cells in the periphery over time. Now, if we look at the myeloid cell expansion, we see that the SGM3 mice have a very robust and very rapid expansion of the myeloid compartment, as indicated by CD33 myeloid positive cells in the peripheral blood over time. But what's interesting, as we notice, is that over time, the overall engraftment of these cells becomes similar between these two models over time. The B cell compartment remains a little bit more robust within the regular NSG CD34 engrafted mice as compared to the SGM3 mice. But the T cell compartment is a little bit more robust in the SGM3. And so this flip-flop between these two may be reflected in just the differences in the percentages of these B cells and T cells within the human CD45 compartment. So if we go back and we look at just the T cell compartment as shown here, if we just look at the CD3 T cell compartment, and we look at within that human T cell compartment the frequency of CD8 positive T cells, CD4 positive T cells, and then a subcompartment of the CD4 T cells called the regulatory T cells. The regulatory T cells are CD4 positive, CD25 positive, and positive for another marker called FOXP3. The regulatory T cells are very important because they are important for controlling and regulating immune tolerance. And as shown in the literature, the NSG SGM3 mice have a more robust regulatory T cell compartment. And that is reflected here within our engraftment model as well. So there are more CD4 T cells, and there are also more regulatory T cells within the NSG SGM3 platform. So of course the next question we wanted to ask was, well, do these mice, these humanized mice, the NSG SGM3 mice, do they also respond to standard of care drugs, and do they respond to immunomodulatory drugs like Keytruda? And here's our preliminary testing of this. This is a breast model that is PD-L1 positive at about 70%. And these mice do have a response to both of these chemotherapeutic agents. They're shown here, and we can see that there's a stabilization of disease or prevention of tumor growth in this platform. Now, we've also tried some combinatorial therapies and some other things here. We've also tried another immunomodulatory agent called Yervoy. Yervoy is a blockage of the CTLA-4 co-stimulatory pathway that is supposed to help release these T cells from energy as well. We can see that there was not quite as robust a response in these particular models, and we've only tried one model so far. This is a lung tumor model, but we do see that there is some significant response. When we do run the statistics on this, we do see some statistically significant responses, particularly when we look at 21 days post-treatment. So we're very encouraged by this, and we're going to be looking at some different combinatorial therapies, and we're going to be advancing our evaluation of these different chemotherapeutics in the future. Okay. So in summary, the NSG are a proven platform for the engraftment of human immune system. PDX growth is not affected by HLA type matching on this platform. The tumor growth kinetics are similar whether you use humanized mice or non-humanized mice. And we've looked at 8 or 9 different PDX tumors to date, and all of them seem to have very high tumor take rates and seem to grow very robustly in the humanized mice. The human immune effector cells infiltrate the human tumors, and this is true both in the human CD34 engrafted NSG mice as well as the SGM3. I didn't show you data for that, but we do have preliminary data showing that there's infiltration of those tumors in that platform as well. The CD34 NSG and SGM3 PDX tumors, when transplanted onto those mice, respond quite nicely, particularly to Keytruda, and we're going to use this as a testing platform for looking at other immunomodulatory and immuno-oncology agents as well. And just to wrap up, I'd like to acknowledge my colleagues at the Jackson Laboratory who did this work. I just have the privilege of presenting this data to you today. The work was performed by several key workers in our in vivo pharmacology services group, headed by Dr. James Keck. Dr. Carolina Paluca, she works at the JAX Genomic Medicine site. We now have a dedicated human genomic medicine site down in Connecticut. She's a researcher who is focused entirely on human genetics and human cancer immuno-oncology, and she's helping us and assisting us with a lot of this data. I would like to recognize Dr. Lenny Schultz, who is the creator of the NSG mice and who has a number of collaborations with folks like Dale Greiner and Michael Brem at UMass Med, etc. So with that, I'd like to say thank you very much for attending, and I'd be happy to answer some questions.
Excellent presentation, Dr. Soper. Thanks for bringing that information to us. Before we get started on all of your questions, here's a quick reminder about how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window. We'll get to as many questions as we can, and we'll give it a few moments. First question is from Yang at Eli Lilly, and the question is, what's minimal N number for the studies?
Okay. Thank you for that question. That's an excellent one. We advise most of our clients that are coming to us doing these kinds of studies to have an N of 10. We realize that this is not an inexpensive model to work with. We do some studies with as low as an N of 8, but we strongly feel for statistical significance that an N of 10 is most appropriate. Remember that these are heterologous tumors, so we do see some variability, and so we do recommend an N of 10 for each of the study arms that are conducted.
Okay, we'll give you another moment. And if there are no more questions, I would like to once again thank Dr. Soper for his presentation. Do you have any final comments, Dr. Soper? Oh, pause 1 second. Hold that thought. Another question from Yang at Eli Lilly. Have you used virus in those models, such as adenovirus or AAV?
Question. Sorry about that. Now I have my voice. Yes, that's an excellent question. We are beginning to get questions about that. There's a number of different things that people would like to do. They would like to do things like use adenovirus or AAV to introduce modulatory agents. Of course, there's things like the CAR T technology, modifying T-cell receptors. People want to do things like label the cells so that they can follow them, expressing molecules like luciferase molecules and things like that. So since this is an earlier area of research, we're just beginning to take on some of those studies right now. We don't have it as an established platform, but we are taking on those studies from time to time, and we're doing what we would call sponsored studies, so that if someone wanted to test those kind of platforms, we're willing to take those on and give them a try. And we have a number of ongoing experiments like that at present.
Tammy from the University of Toledo asks, with the PDX database, is there genomic CNV data available?
Yes, that's a great question. Yes, there is. There's copy number variation data. There's gene variant data. So there's characterization of different specific immune mutations that you can look through, in addition to the gene expression data. So all of that stuff is there. And if there's something very specific that you're looking for that you're not finding, there's a link that you can contact us, and we will try to dig up some of that data for you. We can do some different types of analyses and look for some things. So please get in contact with us if you have more questions about that.
I would like to once again thank Dr. Soper for his presentation. Do you have any final comments to share with us, Dr. Soper?
Yes, thank you. I'd just like to thank the organizers for giving me this opportunity to present this data. This is a very exciting field, and there's a lot of new information being collected. This is an active area of really kind of cutting-edge research. And if you have questions about this, I just want to say that we do have an in vivo services group that obviously is making these mice available to the scientific community. We're also doing studies in-house, where if you do not have the platform established in your mouse, or you do not have in your mouse room, or or if you do not have a vivarium, maybe you're a biochemist or an immunologist that does not have access to the vivarium, we can work with you with these different platforms to test your different agents and do some different experiments with them. So I'd like to thank everyone for joining us today, and I really appreciate the time.
Appears we're having a small technical difficulty there. I would also like to thank you again, Dr. Soper, and I would also like to thank our sponsor, the Jackson Laboratory, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through March 30th, 2016. You'll receive an email from us alerting you when it's available on demand and posted on labroots.com. You're welcome to forward this announcement to any colleagues who weren't able to join in today. Thanks for logging on and participating in today's broadcast. See you next time.